That's actually like the reason why you always see me like waiting till like our picture shows up on the YouTube because at some point I did catch that if I don't wait and I just close the window ooh, once it says it's streaming, sometimes it forgets to like actually do it, even though it told me it would. Okay. So hopefully everybody was able to go through the parts of the heart and get to that activity on some of our heart anatomy, either over break or in the last couple days. And on Monday, you should also have viewed a little bit about the electrical activity of the heart. So we were in this packet and we clipped through basically like the anatomy part of heart activity already. So you took a look at the difference between pacemaker cells and conduction fibers and just like kind of their general different function. We're going to talk in more detail about some of the basis for how pacemakers make a pace today. But then you looked at their firing rates, how they're like kind of connected to each other. And since we were focusing on the spread of excitation, we tracked kind of the impulse and how it spreads between the different conduction fibers of the heart, right? So these yellow lines are all those like pathways. So you wouldn't find them physically in the heart. They're not like a nerve in the way that like your median nerve is a nerve, but as an anatomist, I almost think of them as like analogous to a nerve because they're sending these electrical signals. They're their own thing, they're conduction pathways, but a nerve is kind of a conduction pathway of another type, right? So we know that we have to track them in like a physical space so that we squeeze the heart from bottom to top. Okay, so you went through those steps. So what's new starting for today, if you stopped the video where I told you you could be free and have the rest of your day back, then we left off about here with the ionic basis of the electrical activity. Okay, so when we're looking at this electrical activity in the heart, you don't want different parts of your heart to have like different ideas about when the muscle cells should be contracting, right? Like if you get like scattered contraction throughout your heart, like that's basically a heart attack, right? That's not like really how most heart attacks happen, right? But your heart would not function under that condition. So it's really important that the pacemakers work right. Um, and the pacemakers, right, are literal pacemaker cells, right? So we know we have particularly the SA node in the right atrium and that AV node kind of nestled here near the division between our atrium and our ventricle, right? And we have the pathways between them. Okay. So they're called pacemaker cells because they set your heart rate, right? So they set how your heart beats based on these spontaneous depolarizations, right? So these like automatic potentials that they build, right? So they work without any nervous input from outside the heart. You can make them do this in a petri dish. You don't need to have the rest of any body, right? Like you don't have to have any other types of cells in a petri dish to make these cells beat, which is cool. Um, so they do this in ways that are both similar and different to how we saw like neurons signaling to each other. One immediate difference was that the neurons don't signal to each other out of nowhere, right? They don't do it in the absence of stimuli. They're like sending specific information. But a pacemaker cell is going to do this just kind of on a cycle, right? So think of it like a metronome, right? It's just like clicking back and forth. Okay. So like with neurons, changes to membrane potential, which is what a depolarization is, right? So a depolarization is just moving more towards zero, moving more towards something like an action potential. So the way that we're doing that is we're opening and closing ion channels. So specifically, we're going to see that these spontaneous depolarizations happen as we close potassium channels, right? So remember, potassium usually wants to escape cells. So we stop the potassium escaping and we open two other types of channels. So we know both our sodium and calcium, right? Like the way we set up concentrations of ions in your body, we set it up so that sodium and potassium have these strong electrochemical driving forces telling them to come into cells, right? So as we're changing the state of these channels, whether they're open or closed, right, we stop in some respects the outflow of potassium and we allow in a lot of sodium and calcium. We're going to do these things in a specific order with specific results on like specific like time scales, but that's still the basis of it. Just like sodium and potassium were the basis of our action potentials in nerves, and calcium was a big part of muscle contraction and release of neurotransmitters in our synapses, right? These are same kind of like players. They just have a different job now. Okay. So we're going to see that to depolarize to threshold, like so to actually send an action potential, so like a signal that's going to go through our conduction pathways to the rest of the heart, this is going to be the responsibility of calcium channels, and specifically a type of calcium channel, like a fast calcium channel. Okay. And we're going to need to reset, right? The cyclical activity does reset us, right? Otherwise, we just go up and up and we'd just be stuck in a depolarized state that wouldn't really give you a pace, right? To get a pace, you need to have a signal and then a stop the signal and then a signal and then a stop the signal. So the repolarization part is going to be important too. And that's those potassium channels again. Okay, so let's take a look at the figure. And for the slides in this slide deck, I do want to make sure that you pay attention to the label of what specifically of what type of cell we're looking at. And I'm telling you that because way back when I first taught this course, Dr. Schlater and I try to be doing roughly the same thing, like in parallel. So when I first got these slides, I didn't notice that. And it really makes a difference because we're going to walk through how the heart cells, like the actual like contractile cells work too. And if you don't notice that you're talking about a different type of cell, it gets super confusing because there's a totally different order. The membrane potentials are going to look different. The order in which these ions move is going to look different. Like what's the most important part for these peaks and valleys is going to be different. So make sure you've got yourself oriented to what type of cell we're talking about. So here specifically, we're talking about the pacemaker cells. So we're thinking SA node, we're thinking AV node. Okay. So really should tell you, right? Like the SA node, that sinoatrial node, is the one that is most important, right? Because that's the one that under normal conditions is responsible for your heartbeat because it's the fastest one, right? So it overrides all the others. That's why that's like, if you know someone with a pacemaker, that's like where the pacemaker goes, is like where the SA node should be functioning. Okay. So in terms of how the membrane potential of these pacemaker cells changes, is that we know, like our neurons, they start off low. So they're going to start off around -70, right? And even if we just have them in a petri dish, we're going to see that we have a slow depolarization. This is our pacemaker potential, that automatic depolarization, raising us to a threshold. So still, just like our neurons. Then when we hit this threshold, that kicks off some rapid stuff, right? So just like our action potentials in neurons, we see a rapid depolarization and then a repolarization, and we sink back down, right? It's just, we don't need to have a stimulus for this. There's no signal. They just do this naturally. Okay. So on the part below, what we're looking at is how this links up to the permeability of these cells to specific ions, right? So we can see permeability to potassium and permeability to sodium start first, right? So this first part of the pacemaker cell, right, this first part of the depolarization, we're going up a little bit in terms of our membrane potential because our permeability to potassium is decreasing. So potassium's getting stuck in the cell, and potassium is a cation, right? So that makes us a little more positive. And our permeability to sodium is increasing, right? So sodium, cation, positive ion wants to come into cells. So we're getting sodium coming into our pacemaker cell. So that also pulls us up, right? We've got a little bit of something different going on here in this part before we get to threshold, right? So this is different than what we saw in a neuron, right? In a neuron, when we were talking about the action potential, we didn't see any calcium at all, right? We only saw calcium in the axon terminal when we're ready to release neurotransmitters from a synapse. But in a pacemaker cell and its membrane potential and its depolarization, calcium plays a role. So for this little part here, right before we get to this threshold and set off the action potential, we see here that actually it's not the sodium anymore that's depolarizing us, right? So we can see this permeability to sodium is actually going down, right? So those channels are kind of closing. But making up for it is we have this calcium, right? So these calcium channels are starting to open in this later part of the pacemaker potential. Okay, so calcium is starting to play a big role. And then during this rapid depolarization phase, right, like this fast part, spiking us up, that's all potassium, right? The big thing that's happening here is unlike in a neuron, right? In a neuron, we had sodium rush into cells. Here we have calcium rush into cells. Calcium is really driving the pace of your heart, right? So if you know anyone with heart issues, right, they may be getting like specific directions on how much calcium they should get in their diet, how much salt, so sodium, should they get in their diet, right? That's partially to do with the fact that these ions are really important to heart function, right? Partially that, partially also like about the effects on like blood pressure and things like that. But we won't talk about that part till A&P 2. So for now, I'm just going to make the point that ions are important to the heart. And so calcium increases a lot here. Okay. So then as we repolarize, we can see that that is due to the calcium channels closing, right? So that permeability to calcium is decreasing here, right? And we are opening back up those potassium channels, right? So potassium is now leaving the cell again. So it's escaping, pulling us back down to quote unquote normal, right? But really for our pacemaker cell, that's just the valley, that's just the dip. It's not like we sit here for a long time, right? It's not like our resting membrane potential like it was in a neuron, right? Because as soon as we go down, we start to climb back up because this autorhythmicity of the heart of these pacemaker cells. Good. So now is when I'm going to key you in. We're switching to a different type of cell, right? So these were our pacemaker cells. So setting the heart rate. Now we're thinking about contractile cells, right? So our contractile cells, you're thinking like the muscle cells, right? The cardiac muscle actually contracting, ultimately actually, right, like squeezing the blood out. So those cells work differently. They still are going to have like action potentials, right? They're still going to have this change in electrical activity, this change in permeability to ions, change to membrane potential, but it looks different, right? They're going to be using these action potentials in their contractile cells to contract rather than to send signals out to the rest of the heart. So in the contractile cells, we can divide it into 4 numbered phases, except for really it's 5 phases because we're 0 indexed. which you may or may not have talked about that if you've never taken a computer science course, which I expect most of you probably haven't, right? We start counting with zero here, right? So we're going to go 0, 1, 2, 3, 4 when we're talking about the phases of electrical activity in cardiac contractile cells, so those heart muscle cells, basically. Okay. So you'll notice that the steps are a little different than we went through on that last diagram. So we can see that the starting phases here, right? We kick it right off the bat with sodium, and we don't see potassium really playing a role until later on, right? So we see that in phase 0, this is all about permeability to sodium increasing. In phase 1, we're actually leveling off that permeability to sodium. So we're turning off that permeability to sodium. But we have a trade-off, right? We then have another ion that usually wants to come into cells. So that's our calcium, right? So our calcium is coming in. And it's when we let the calcium in that we kind of turn off that potassium escape. And then in phase 3, we let potassium escape anew because we increase the cell's permeability to that. And we're turning off this flow of calcium before we get to a resting membrane potential at the end of these cardiac contractile cells and their phases of electrical activity. Okay. Um, so overall, we're going to see that the action potential of a cardiac contractile cell is a lot different than you usually see in skeletal muscle, a lot longer, right? So it's on the order of 250 to 300 milliseconds compared to a skeletal muscle is only like 1 to 2 milliseconds. So difference. Okay, so here's the graph that we were talking about. Okay, so 0 actually starts with this green line going straight up, right? So kicking off the activity of our cardiac action potential, right? So we're thinking cardiac contractile cells, right? This is why I'm saying pay attention to labels. Is this graph has the same colors and it's got numbers and it's got steps and it's got ions, but super different looking. Okay. So membrane potential of the cardiac contractile cells. Starts out phase 0 with a huge spike, huge depolarization, and that's all sodium rushing in. So that's our sodium permeability. Then we have a short slump, right? So this phase 1 is caused because we cut that sodium off, right? So we let in a ton of sodium, then we decrease the permeability of the cell to sodium. So we see a brief repolarization, right? So a brief kind of sink and slump. And then in phase 2, while we're starting to let some calcium in and we're starting to decrease how much potassium is escaping, we see kind of a flattening out, right? So we're kind of reconfiguring during this phase 2 with this kind of plateau before we sink a little further down, right? Like ultimately, as we're doing these things, this does bring us a bit lower. It's just a bit slower than phase 1 in this repolarization. And then in phase 3 is where we see our big repolarization, like most of getting us back to rest is as we close our calcium channels and we open the potassium channels, right? So that is the repolarization phase, is blocking those calciums, those positive cations that were coming in and letting our potassium leave the cell, so pulling a bunch of positive charges out. And that's how we get back to rest. So we are going to have to couple up excitation and contraction in the heart, right? So we have to link up like electrical signaling to actually contracting cells. Okay. Um, and we're going to see that because we're thinking about heart muscle now, right? Like cardiac muscle is a little bit like skeletal muscle and it's a little bit like smooth muscle, right? So our cardiac muscle is going to be striated, but it's going to be involuntary, right? So we know like big picture, some things are different between our three groups of muscles. Now we're getting into a little nitty gritty kind of stuff on our cardiac contractile cells, our cardiac muscle. So when we think about what's similar to excitation and contraction, so like muscle activity on a detailed physiological level to a skeletal muscle, we're going to see that just like in our skeletal muscle, cardiac cells have T tubules. So those were those like tunnels diving into our muscle cells in skeletal muscle. We'll see that we have sarcoplasmic reticulum storing calcium. So these were like these sacs, basically, like these bags of calcium that are a modified version of our endoplasmic reticulum. So you'll still see those in skeletal muscle. And if we were to look at the details of how the contraction is actually happening, like, so how do these fibers get shorter? How do we have things slide past each other? We'll see that that activity is still regulated by the interaction of calcium with troponin, which we think of as like kind of our light switch for muscle contraction, and the tropomyosin, which was that long skinny thing kind of covering up our binding sites that are blocking actin and myosin from grabbing onto each other in the absence of calcium. So that's all going to be similar to skeletal muscle. I would say in general, like heart muscle looks more like skeletal muscle, at least as far as this process we're about to talk through goes. Um, what's going to be similar to smooth muscle instead, so this muscle that we see in like your intestines or your blood vessels, we're going to see that there are gap junctions. So we talked about that with the single unit smooth muscle, right? Like one of our types of smooth muscle that we mentioned before break, right? So this means that the heart is going to be able to like coordinate a little faster, a little better, which is good because you want the heart coordinating. That's kind of the whole point. If you've got signals bobbling around and trying to activate each other in weird orders, you are not going to get a good heartbeat. You are not going to pump your blood around your body effectively. And another thing that's similar to smooth muscle, which is a detail we didn't really go into about smooth muscle, um, but not all of the calcium that we're looking at is actually already inside the cell in skeletal muscle. It was really pretty much all already in there, stored in the sarcoplasmic reticulum when we were thinking about where does that calcium come from that lets us start cross-bridge cycling. In smooth muscle, some calcium comes from outside the cell, like you actually pull some in, and that's going to be true for a heart cell too. So this is our big picture of how it works, which we can step through, but I'm just going to point out some of those features that we mentioned in the previous slide while we have the whole thing in front of us, right? So here you can see that T tubule that should be familiar from our skeletal muscle cells. Here you can see that sarcoplasmic reticulum that's similar. Here you can see calcium coming from sarcoplasmic reticulum that's similar. Here we have our nice, familiar, hopefully cross-bridge cycle happening in the cell. We've already talked through all that when we talked about skeletal muscle. What's going to be different is this calcium stuff happening on the surface of the cell, right? So on this plasma membrane, right? So we're going to see this relationship between calcium and the outside of the cell. And we can see this gap junction, right? So we can see that the neighboring cell, right, the cell next to it, if it has an action potential, whoop, it can shoot through and send that action potential to the neighboring cell really quickly. We don't have to worry about receptors. We don't have to worry about time lag when we're trying to get our heart cells to contract together. So how would we do this in terms of steps? We'd start out by sending an action potential through this gap junction, right? So this is our straight shot into a neighboring cell. This cell is contracting or is like a pacemaker cell and is going through these spontaneous depolarizations, giving us still a spike of depolarization, right? Like, which is an action potential, right? So either way, we're getting the signal depolarizing the cell we're looking at, right? So this current voltage is spreading into our cell here. Yep. So then that spreads out, right? So it gets to the rest of the cell. So here you see that action potential traveling along the membrane and also diving down into the cell through this T tubule. So we saw that part before in our skeletal muscle. Okay, so here's one of the different pieces. So after we spread this action potential out, right, we're now trying to get calcium into the cytoplasm so that we can do our cross-bridge cycling. So some of that calcium is coming from the extracellular fluid. So some of the calcium is coming here through our calcium channels on the plasma membrane that faces the extracellular fluid. So that's not like skeletal muscle, but some of that calcium is coming from the sarcoplasmic reticulum, which is like skeletal muscle. So we have more ways of getting calcium. That's great. We got like backup. Okay. So then we have our cross-bridge cycling. So we see our calcium binding to troponin, that little pink snowman. So this is our light switch for turning on the myosin binding sites on actin. So this blue-purple guy is our actin. Those like olive pits are the myosin binding sites on the actin so that the actin and myosin can hold on to each other. All right, so here we see our cross-bridge cycling. So this is when the cell would actually be getting shorter, aka contraction. All right. And then we have to relax. So in order to relax, in skeletal muscle, you just put the calcium back into the sarcoplasmic reticulum. So we do see that happening. That's one part of relaxing your heart cell. But the other thing is that you're going to pump some calcium back to the extracellular fluid too. So here you see a calcium pump, right? We know it's a pump. We know it's primary active transport because we see this use of ATP, right? And here we see some secondary active transport happening too, right? So you can see we're using sodium to counter transport some calcium out of the cell as well. So we've got like two steps to relaxation here. So this relaxation can happen like kind of fast, right? We got two ways to do it. Okay. Once the calcium's gone, we go back to our relaxed state, which means we don't want to be doing our cross-bridge cycling. We don't want to be contracting anymore. We want our cells to loosen up. So we close back over to our original state, these actin molecules. So relaxation of cardiac muscle is super important because imagine your whole heart just like stayed clenched up. You would die. That would not work. That would suck, right? So we need to remove calcium from cytosol. We saw it in the picture form on the previous slide. We see it here in words, right? So we have our calcium ATPase, aka a calcium pump, on that sarcoplasmic reticulum, pulling calcium into this storage space. We've got a calcium ATPase, again, a calcium pump on the plasma membrane, putting some calcium out into our extracellular fluid, so just eliminating it from the cell. And we have this sodium calcium exchanger, that secondary active transport also going on at the plasma membrane. And we are fully relaxing because once there's no calcium, troponin and tropomyosin go back to their spots, right? They go back to their relaxed states. We'll do these questions on Friday when we're in person as some review stuff. We've got still a little bit of time, so let's talk about electrocardiograms or EKGs. Okay. So they're EKGs because in German, if you were to spell electrocardiogram, you would spell cardio with a K, and that's how we name them. But sometimes you'll see them as ECGs too, right? Because we don't speak German. Same thing, right? So whatever you see it called, they're talking about the same thing. These are great because we can record electrical activity from the outside of your body. We do not have to physically dig into your heart and figure out what every individual cell is doing to know whether the heart is doing it what it's supposed to do, whether its action potentials are happening when they're supposed to happen, whether all this conduction is happening normally, right? So it's non-invasive, right? You do not have to do a surgery to do this. You don't even like need any particularly complex machinery. I guess as a non-engineer, I shouldn't say that. I don't know how hard it is to build one of the machines that can do this. But it's pretty easy to run. So we can do this from the outside of the body, and we do it by putting electrodes on a number of specific places on the skin. And your body like physically conducts these electrical signals, so you can sense them with the electrodes. And then the machine does a bunch of math, right? So a bunch of like triangulation, basically, to figure out what's going on with the electrical signals in the heart. So this is how you would test, does someone have a problem with the conduction of these electrical signals, and what is that why you're seeing some specific heart malfunction, some problem here? Okay. So this electrocardiogram is an external measure of electrical activity of the heart. It does require multiple points of contact, so multiple electrodes. You can't just like put one on top of the heart and immediately know everything that's going on. It uses this principle that your body can act as a conductor. Um, it can, right? This is part of why you don't want to get struck by lightning. This is part of why you don't want to stick a fork in an electrical socket, right? Like electricity will run through your body, right? Um, when we've been talking about conduction, we've been focusing on like when your body does it down specific routes on purpose, like in nerves and neurons and these conduction fibers in the heart. Um, but there is like radiation out of this stuff, like even up to your skin. Yeah. So these currents, this is also like why you don't drop a toaster in the bathtub, right? Like your body is mostly water. Currents spread really easily through water. Um, so they can spread to the surface, and this is a fact, a principle that we use for other types of recordings too, right? So we use them for ECGs or EKGs, right? Like measuring heart function. But you'd also use them for measuring muscle. You also use this for measuring brain waves. This is useful. This is useful for all kinds of recordings, interpreting all kinds of function, a way that we can understand what's happening inside the body in a way that's difficult to like physically see, but we can figure out by doing some math. Okay. So the math that you would do, we will not be going through any actual calculations here. I don't actually know the actual calculations, but we do want to understand like what the basis of this is, right? So one of the things that we are going to keep in mind when we're using these electrodes to record, right, is that how far these things spread, so the distance they spread, and the amplitude, right? Like, so how big these spikes are in this spread is going to depend on the size of the potentials that are actually happening in the heart, right? So, like, how big were the spikes if we're looking at those depolarization in the pacemaker cells? How big are the spikes if we're looking at the action potentials in the contractile cells, right? So that would make a difference. We can get some information about peaks, or we'd be using the fact that they have specific peaks. And it's also going to depend how many cells are doing this at once, right? Like if just one cell is depolarizing, you're not going to be able to sense that from all that far away. If every single cell in your heart is having a depolarization at the same time, you can super sense that, right? Like that's going to be spreading much farther, and it's going to look much bigger at the places you're recording, right? So that's a piece of how we link together what's actually happening in the heart to the math we do to figure it out as we're doing this electrocardiogram. Fortunately, right, like you can put these electrodes on the skin, not all directly, right, exactly on top of the heart, because your heart cells do synchronize, right? Like they do have their depolarizations basically at once. You're not trying to find the electrical signal of just one teeny tiny cell. We got a bunch of them doing the same thing at once. So these things are big enough to record, basically. So here's what those things look like. All right. So we are looking here at the membrane potential of one of those cardiac contractile cells, right? So we talked about those phases of the membrane potential of the contractile cells, right? We can see that. So that's what's happening physically to the cell's membrane potential. But here's what your recording would look like, right? So the recording is based on where you've put electrodes on the surface of the body, and they're doing like this math about distance and amplitude and peaks and stuff to make this probably more familiar thing, right? You've almost certainly seen one if you've worked in a healthcare setting in some type of clinic, but you've also seen these like on TV, right? Like you see these things in TV shows all the time when something dramatic in a hospital is happening. So this is what the recordings we read as clinicians ends up looking like. So these different peaks and valleys correspond to different parts of the electrical activity of the heart, because when we say, right, that heart electrical activity is synchronized, it is like stuff is working together all at once. But as we're about to see in the cardiac cycle, right, we have four chambers of the heart, right? We want blood to go from the atria to the ventricles. You saw that when you were looking at kind of the order of blood flow in the cardioanatomy piece, right? So it's not every single heart cell depolarizing at the same time, right? This is happening kind of in a wave through the heart in a specific order. All right. So we can tell things when we look at this electrocardiogram about, okay, is it an atrium that we're looking at contracting versus is it a ventricle that we see contracting? Or are we looking at repolarization? Are we looking at going back to a relaxed state, right? So all of that information is in this electrocardiogram, right? So you would be able to tell, like, oh, is this a problem with atria? Oh, is this a problem with like relaxation and repolarization? And is that in the atrium or is that in the ventricle? Or does it have to do with like signaling between our different kind of conduction pathways and the SA node to the AV node, things like that? Right. So there's a lot of stuff you can solve basically with an electrocardiogram, a lot of different pieces of heart function you can look at here. Okay. So we'll be talking about some more of this as we go through the cardiac cycle. But linking these up for right now, right? Fundamentally, this little peak here, this P wave, this is going to be when our atria depolarize. So that top part of the heart, right? That part where we have venous flow coming in. So stuff is coming from elsewhere in the body into the heart. That's the part we see depolarizing first, right? You want to squish that down into the ventricles before the ventricles contract, right? Because you want all, it's not going to be all the blood, but like you want all the blood to be there when you squeeze it out to the body, right? So that's our P wave here, is this little peak. Then we're going to think about our QRS complex, right? So we've got Q, which is this valley. We've got R, which is this peak. And then we've got S, which is this diving down here, right? So we're thinking about this kind of all together. So what's happening here, right? We already had our atria depolarize, right? So we're thinking about the atria squeezing blood into the ventricles. And then next in this complex, we've got the atria relaxing, but we've got the ventricles depolarizing, right? So here we've got relaxed atrium, but our ventricles need to squeeze, right? So big peak, right? before we end up going back to ultimately our ventricles repolarizing. Right. So this is the depolarization of the ventricles and the atria going back to normal. And then it's the T wave. So this little hump, that's our ventricles repolarizing and going back to relaxed and back to normal. This means that since we know what's happening in these general places, right, P, T, and this QRS, we can measure other things, right? Like other parts of like how long is your heart squeezing? How long is your heart relaxing? By measuring how long it takes to get between these points, right? So for example, between P and Q, this is your AV nodal delay, right? Which makes sense, right? Like this is how long it's taking us to get this signal from the atrium to this atrioventricular node, and we got this pause so that the ventricles can do their thing and get ready to squeeze. More importantly, actually, are these ones in red here, right? So our QT interval, so between Q and T, it's labeled on the right side, but actually I'll just point them here, right? So this dip to this peak, this is ventricular systole. So when we go through the cardiac cycle, we're going to be focusing on the ventricles because they're kind of the most important part of the heart in that, like, that's what actually gets the blood to your body, right? All the parts of the heart are important, but like the point is getting blood to your body. So the cardiac cycle is going to be dependent on the ventricles, is what we're going to be tracking. And ventricular systole, systole, think squeezing, right? So ventricular systole is going to be when are your ventricles squeezing? So that's going to happen from Q, right, where we have these ventricles depolarizing, all the way to T, where we have those ventricles repolarizing, right? So from Q to T, we got squeezing the heart, all right? So then from T all the way back to Q on our next little section of our electrocardiogram, this long flat part, that's ventricular diastole, right? So you'll notice it's longer, and we'll talk about that as we talk about the cardiac cycle, but that's the relaxed part of your heart. And you want it to be long for a bunch of reasons, right? Like one is that you want the heart to have enough time to fill with blood. But another is you don't want your heart to work too hard, right? Like think about like how uncomfortable it is to have your heart rate really fast. There are other reasons having to do with like nerves and muscles that like it's kind of uncomfortable to have a fast heart rate, but also your heart is kind of working too hard, right? Like hearts actually can get tired. You want them to have enough time to kind of rest. So that's that TQ segment. Okay. All right. Um, we'll intro the cardiac cycle a little bit. So overall, we're going to go through numbered phases. All right, so we saw those numbered phases earlier today in our cardiac contractile cells. Unfortunately, those are totally different numbers and totally different phases. You should know them, but the ones we're about to talk about in the cardiac cycle are like top level what you want to study, right? The phases of the cardiac cycle are way more important. These ones that we're about to talk through in this slide set. So if you're prioritizing what to study for the exam, it's these phases, right? Like they're, might be a question or so on the other phases having to do with those ions, but there's definitely going to be a bunch of cardiac cycle stuff because this is how you get blood going to your body. Okay, so we're going to go through the phases of the cardiac cycle. And in order to get blood to flow in a specific order through the chambers of your heart, which we need to do, right, to get blood going through the appropriate tubes, this is actually going to all be dependent on pressures. Now, usually when I pull the class, I forget if I've pulled the class yet this year. Usually people have not taken physics before this course. That's fine. We'll talk about the concepts. If you have taken physics before this course for some reason, great. Like think about pressures, right? You might also have seen some of this stuff in chemistry. I haven't looked at the chemistry syllabi in a while. We're going to be thinking a lot about relationships, specifically between pressure and volume, right? And if you haven't taken any of these things ever before, that's fine. I'm going to explain them to you, but these are things to pull back to your memory if you do happen to have seen them before. Okay. So we're going to be thinking about pressures in our chambers of the heart, the atria and the ventricles, and also in our big, main, most important blood vessel, the aorta, that comes out of the left ventricle. We're going to be thinking about volumes of blood, right? So we're going to be thinking about pressure on that blood, like how hard are you squeezing it? And we're also going to be thinking about how much blood there is. And then at the end of this, we're going to link this up to like, if you put a stethoscope on someone's chest and you're listening to the heart, like how does this link up to all of that? Like what are you hearing? When should you be hearing it? And when they listen to your heart and go, "Uh-oh," what did they hear? What part of the heart cycle is that and what might you be able to tell from it? So that's kind of our goal as we go through cardiac cycle. So the whole cycle, what we're going to be tracing through is the blood and how it goes through your heart during a single complete heartbeat. So like the boom, boom, right? That's what we're going to be thinking about as we go through the cardiac cycle. We're going to be explaining all those pieces. The reason I'm saying we're starting introing it today, even though there's a lot of material in this overall packet, is that a great part to start and something that I think you should be able to fit in your brain in a minute or two because it's not so bad, uh, are these concepts of systole and diastole, right? Like I just mentioned them on that electrocardiogram. Right. So we've got a contraction phase, right? So squeezing, systole, right? I like that all the S words kind of go together in physiology. Squeezing, systole, sympathetic, stress, right? Like all these things are actually kind of coordinated, as we'll see, right? We even will come back to some sympathetic nerves later on, right? Squeezing, systole, ventricular contraction, right? Squeezing down. Diastole, those letters don't match up. It's just the opposite, right? So if we've got our squeezing contraction part, we're also going to have the relaxation part. That's the diastole. Okay. These do match up to if you've ever taken a blood pressure, right? You've got a systolic and a diastolic pressure, right? Like what they're measuring is like how much is the pressure in your blood vessels when the heart is squeezing versus how much is the pressure in your blood vessel when your heart is chilling out, right? Like how much pressure do you have when it's relaxed? You're going to have some blood flow, some blood pressure regardless, which is good. You want blood getting to your tissue all the time, but it's not getting there like as strongly in different parts of the heartbeat. Okay. So the other thing that we want to know in order to prep ourselves for our discussion of the cardiac cycle is we want to remember about valves. So I want you to realize like kind of where they are in the heart. But the thing that we want to know is that valves open basically because someone's like pushing on a door, right? So an AV valve opens when the blood in the atria is pushing on the door of the AV valves. So that lets us go into the ventricle where we've got kind of a vacuum situation going on, right? So we go from high pressure to low pressure, just like we go from high to low with every gradient. Semilunar valves are different in shape, a little different in function, but again, we're going to see these relationships between high pressure and low pressure. Blood is always going to flow from high pressure to low pressure. Okay. So I know we're at time, but that's great because I think this was a lot of information. Next time we'll go through all these different phases of the cardiac cycle. So if you want to familiarize yourself with some of the terms, it'll probably be easier and more relaxed to follow next time. But if not, I will see you on Friday. All right. Take care, everybody. Whoop, what we got here. Oh, So we had a little bit about Twitch stuff on the previous exam. So we will have some muscle stuff on this exam that we didn't get to, but we'll mostly focus on the fiber stuff. At most, if there's going to be Twitch stuff, I would expect that might be something like an extra credit question or something. Maybe one is kind of the answer on that.